This unit looks at how we take half of the DNA from one parent and half from another to create a fertilized egg and subsequent zygote. So we will be focusing on the germ cells, not the somatic cells or body cells that we just finished discussing with mitosis. To get us started, we will look at a chromosome, which is a highly wound strand of deoxyribonucleic acids, or DNA. You will also see that this highly coiled DNA, or chromosome, is organized into 22 pairs of what are called autosomes, and one pair of sex-linked chromosomes, for a total of 23 pairs of chromosomes. Key point is that what we find is in each somatic or body cell. The arrows are showing you what we call homologous chromosomes, or the nearly identical pairs. They have genes lined up on their DNA in the same relative order with some variations or variants for certain genes. The map you see on the right is a karyotyping of a typical diploid, or 2N, somatic cell. Comparing the pair of 23rd chromosomes, or sex chromosomes, we find that in this table, males have an X and a Y, whereas females have two X's. The size of the male Y chromosome is much smaller than the female X chromosomes and contains very few genes. Indeed, you can see this in the table. The most interesting feature of the Y chromosome is that the SRY gene will help create a male reproductive tract. The female has a much larger pair of X chromosomes, but not a clear standout like the SRY has been yet found. Building on the difference between gametic and somatic cells, you can see that our gametes, namely our sperm or egg, have only one copy or are haploid of those chromosomes. These are the only two types of gametes found in humans. The sperm and the egg come together to form a diploid or 2N fertilized egg and subsequent zygote. The somatic cells are all the other diploid cells in the body, including skin, nerve, muscle, and bone, to name a few. Remember that all gamete cells are produced by meiosis, whereas all somatic cells are produced through mitosis. The meiotic process creates our gametes with the requisite half of all chromosome numbers, and these occur in special cells called germline cells. The key to meiosis and its connection to sexual reproduction is that it ensures that there is genetic variability and that we end up with haploid cells to allow diploid zygotes. This occurs through three key processes: independent assortment, crossing over, and random fertilization of different sperm with different eggs. Also, you'll note that when the chromosomes get close together, they can exchange some sequences from another called crossing over. Also, you will note that when the pair of chromosomes are close to each other, they form a structure called a tetrad. Tetrads are very important in the first meiotic division in order to maintain the diploid state of the two daughter cells and to aid in the crossing over event. Meiosis consists of two divisions, where we only saw one division when we looked at mitosis in somatic cells. The terminology for meiosis is that the first cell, which is diploid, is called the parent cell. The resulting cells are called daughter cells. The first meiotic division reduces the number of chromosomes in half from 46 to 23. Then, in the second meiotic division, the two separate cells go through a further division to generate 4 haploid cells with only one copy of each chromosome. So summing up, the first meiotic division reduces chromosomes in daughter cells with the half, or 23. Then through the meiotic division 2, it's more like mitosis. We end up with 4 haploid cells with 23 chromosomes. You can watch this animation of the meiotic process or skip to a more detailed explanation in the subsequent slides. This overview slide of Meiosis I shows that the parent cell goes through 4 stages namely prophase I, metaphase I, anaphase I, and telophase I. During the prophase, the chromosomes come together and spindle fibers attach to the chromosomes. Then the chromosomes line up at the middle or equator of the cell in metaphase I. In the anaphase I, you will see the chromosomes separate and move to the poles of the dividing cells. And by telophase I, you will finish cytokinesis and the two haploid daughter cells will move into interphase. Here we see that the prophase I is a process where the cells are allowing the chromosomes to condense or come together. The spindle formation helps align the chromosomes, and in the final stages, the nuclear envelope breaks down to allow eventual cytokinesis or cell division. During the prophase I of meiosis I, the chromosomes line up and homologs of genes cross over from one chromosome to the other. Please note in the picture that you can see that the gene A from green has crossed over and becomes part of chromosome blue, whereas the A from the blue chromosome crosses over to the green chromosome, and this allows the genetic variety that we have discussed. Indeed, with an average of 8 million different combinations of chromosomes, there are more than 70 trillion genetically unique combinations possible, meaning that cells will be able to make almost limitless combinations in genes. Now the homologous pairs of chromosomes pair up along the middle of the cell. But here is an important second step in genetic variability, namely independent assortment. How the green and blue chromosomes line up in the middle is random. You can see the order of the chromosomes of green and blue lead to 4 different combinations of haploid daughter cells in meiosis II, depending on how the homologous chromosomes line up. Here you see the sister chromatids remain attached at their centromeres and move to the opposite ends in anaphase I. In the final phase of meiosis I, we see that the nuclear envelope that disappeared in prophase I comes back and the spindles disappear. This allows the actual division of the cells, called cytokinesis, to result in the 2 separate haploid daughter cells. Between the first meiotic and second meiotic phases is an intermission of sorts. This short time period allows an interphase for chromosomes to fold, proteins to be made, and the key point here is that DNA is not replicated. The process of the second division is what meiosis II is all about. Here you will see that the series of 4 steps is the same, but this time there are 4 cells that come out with only haploid or one end of chromosomes. Remember, we are discussing the gamete cells only here. Much like we saw in meiosis I, here in the first 2 phases of meiosis II, we see the chromosomes condense, spindles form, and the nuclear envelope disappear in prophase II. Then they line up in the middle in metaphase II. Then the cells go through a migration to the opposite poles and finally split the cells into 4 haploid cells in telophase II respectively. The amazing process of meiosis results in 4 haploid gametes with a unique assortment of genes and chromosomes. You can see in this chart that mitosis and meiosis differ in many ways. Key is that mitosis is only 1 division, where meiosis is 2 divisions. You also see that mitosis gives you 2 daughter cells, where meiosis gives you 4 daughters. Mitosis is setting up to make carbon copies, so they result in identical cells, whereas meiosis gives you unique or genetically different cells. You also remember that mitosis only occurs in somatic cells, and meiosis occurs in genetically gametes, or sperm and eggs. The big picture is that we use mitosis for repair, growth, and asexual reproduction, but meiosis is only for generating new gene combinations for sexual reproduction. You can watch this video on meiosis versus mitosis at this point. Once you have the haploid gametes, or sperm and ovum, they must come together in order to generate new life. Sperm travel to the egg and use enzymes in their heads to break into the eggs. Most of the time, only one sperm makes a successful entry into the egg because the egg has a way of blocking multiple sperm entering. Remember that we join the haploid egg nuclei with DNA with the haploid sperm nuclei with DNA to form a diploid zygote. When fertilization takes place, there are some things that can happen when one sperm fertilizes one egg and an early division leads to a shared placenta. These embryos are called monozygotes, or commonly referred to as identical twins. They share one placenta. The other possibility is that these two simultaneous fertilizations with two separate sperm that lead to two different embryos with separate placenta called dizygotic twins. This is more commonly referred to as fraternal twins. In the prior slides, we discussed normal meiotic principles, but sometimes there are errors in meiosis where chromosomes do not separate, called non-disjunction. Non-disjunction literally means not come apart. This leads to an abnormal number of chromosomes in our final cells. One example is called trisomy 21, where there end up being 3 chromosomes for the number 21. This is more commonly referred to as Down syndrome. Trisomy 21 is the most common form of mental retardation, and maternal age leads to higher risks for non-disjunction to occur. Indeed, in women that are 44, there is a 1 in 30 chance compared to a 1 in 1,530 chance in a woman having a child in her early 20s. This completes Meiosis I unit.